Thank you, Brenda, for your kind introduction. Hello, everyone. My name is Govindaraj Kumaran. I'm a CMC reviewer in OPQ, Office of Pharmaceutical Quality, CDER FDA. So I'm happy to be here to present nitrosamine risk assessment in type 2 DMFs supporting GDUFA applications. A quick disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. I would like to start off with OPQ's overall goal and mission. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. This slide provides the learning objectives of my presentation. First, I will discuss the formation of nitrosamines, and then nitrosamines in drug products and FDA guidances, potential root causes of nitrosamines in API, nitrosamine risk factors in API, recommendations to API manufacturers, approaches to determining acceptable intake limits, nitrosamine control strategies, and finally, summary. So the nitrosamine is a class of compounds in which the nitroso group is attached to an amine. One of the most common way to form the nitrosamine is the nitration of secondary amine under acidic condition. If a nitrosonium species is generated from the nitrosating agent, then the secondary amine will readily react with the nitrosonium species, and it will produce the nitrosamine. Tertiary amines may undergo dealkylation to form secondary amine, which may undergo nitration to form the nitrosamines. And amide solvents may decompose to secondary amine, which may undergo nitration to form the nitrosamines. So the nitrosamine is a probable human carcinogens. They are potent genotoxic reagents, and the nitrosamines are referred as a cohort of concern in ICH M7 guidance. Because of their high carcinogenic potency, they are regulated more tightly than typical mutagenic impurities. The TTC limit, that is a threshold toxicology concern limit, should not be applied to the nitrosamines. This slide describes the small molecule nitrosamines in drug product. The first nitrosamine issue was reported in June 2018. So in June 2018, FDA informed the presence of NDMA, that is N-nitrosodimethylamine impurity from a valsartan manufacturer, and then later small molecule nitrosamines in drug product were reported, and some of them are listed here. Then to address these small molecule nitrosamines, FDA first published the nitrosamine guidance in September 2020, and then it was later revised in February 2021. The guidance is control of nitrosamine impurities in human drugs. So this guidance actually provides some basic information about the nitrosamines, and also it provides a control strategy for the nitrosamines in drug substance and drug product. More importantly, this guidance recommended control limits for certain small molecule nitrosamines. Some of the small molecule nitrosamines are listed in this table. At the time, these small molecule nitrosamines are probable nitrosamine impurities in the drug substance and the drug product. When the small molecule nitrosamine impurities were reported in the drug products, simultaneously, NDSRIs in drug products were also reported. NDSRIs are nitrosamine drug substance related impurities. They are class of nitrosamines sharing structural similarity to the API. FDA identified this issue in a public statement on November 18, 2021. So the presence of NDSRIs in drug substance and drug product added additional complexity to this nitrosamine issue, primarily because determining the acceptable intake limit for these NDSRI is more challenging than the small molecule nitrosamine impurities, primarily because these the NDSRIs are unique to each API, and there is no safety data or carcinogenicity data reported for these NDSRIs. So determining the acceptable intake limit for these NDSRIs is more challenging. So that becomes a huge issue in the pharmaceutical industries. To address this issue, FDA again published one more guidance in August 2023. That is recommended acceptable intake limits for NDSRI, that is rail guidance. So the major component of the rail guidance is the CPCA, carcinogenic potency categorization approach. Using CPCA, the rail guidance categorizes the NDSRI into 5 different categories, and the associated acceptable intake limit ranges from 26.5 nanogram per day to 1,500 nanogram per day. You can find this guidance in this web page, and the link is provided here. And this web page also provides some additional information on acceptable intake limits. There are 2 tables are there in this web page. Table 1 contains searchable list of 264 hypothetical NDSRIs, potency categories, and associated recommended acceptable intake limit. And the table 2 contains list of NDSRI for which FDA has identified acceptable intake limits based on compound-specific data or read-across analysis. This web page will be updated periodically. So moving on to the next section, root cause of nitrosamines in API. The root cause of nitrosamine in API can be categorized into 3 categories. First is intrinsic properties of API, process-related, quality of raw materials. Intrinsic properties of API. Suppose if an API has a nitro group and a secondary or tertiary amine in the same molecule, then there is a possibility that the API can generate the nitrosamine impurity. For example, in the case of the ranitidine, this ranitidine has nitro group and a tertiary amine in the molecule, and it was reported that the ranitidine can generate the NDMA impurities by the intermolecular degradation of API without the requirement of any exogenous nitrate source. So the other category for the root cause of nitrosamines in API is process-related. In the most common pathway to formation of nitrosamines, 3 factors are required: presence of nitrosamines, presence of nitrosating agent, and conditions conducive to nitrosamine formation. Presence of nitrosamine, nitrosable amines. Excuse me, let me have some water. Okay, presence of nitrosable amines. Amine functional groups are very common on API molecules, isn't it? Almost 20% of the approved drug products contains the amine functional groups. And the tertiary and secondary amines are common bases used in the synthesis. And amide solvents are very common in the API manufacturing process. They can significantly degrade under high temperature to form secondary amine. For example, dimethyl formamide. Dimethyl formamide can degrade at high temperature to form the dimethylamine, and that amine could cause the formation of the nitrosamine impurities. So the other factor for the formation of nitrosamine is the nitrosating agent. So the nitrosating agent can be intentionally added as a nitrate salt or nitrous acid, or It can be an impurity from another reagent, such as nitric acid or sodium azide. The less common nitrosating agent is alkyl nitrates. Alkyl nitrates may be intentionally added to the API manufacturing process, or it may be an impurity from a rearrangement of nitroalkanes. So the third factor is the conditions conducive to nitrosamine formation. Acidic condition facilitates the nitrosating reaction. So the other process-related root cause of nitrosamine is the oxidation of 1,1-disubstituted itrazine. In the case of 1,1-disubstituted itrazine, you don't really need any external nitrosating agent. Even the molecular oxygen can oxidize the 1,1-disubstituted itrazine, and that will generate the nitrosamine impurities in the API manufacturing process. Nitrate formation from oxidation of hydroxylamine is known, or nitrate release from nitroaromatic precursors is also known. And the other root cause of process-related root cause of nitrosamine in API is fluid bed drying. Nitrogen oxides in the intake air of the fluid bed drying can generate the nitrosamine in the API. And use of activated charcoal also can pose the nitrosamine risk, because when you use the activated charcoal in the final stage, especially in the final stage, and if there is a secondary amine present in the process, then there is a risk for the formation of the nitrosamines. So the other process-related root cause of nitrosamines is a poor operation design of the process. Suppose if nitrosamine is formed in the API manufacturing process, and because of the poor operations or design of the process, the adequate purging may not happen for the nitrosamine impurities. Then that impurity will carry over to the final API product. And cross-contamination of nitrosamine impurities in multipurpose facilities has been reported. So the other category in the root cause of nitrosamines in API is quality of raw materials. Use of raw materials containing impurities. So the amine and nitrosamine impurities were found in the fresh solvents. Some of them are listed here. And the use of disinfected water can generate the nitrosamine impurities. And nitrate impurities are found in the several reagents, such as sodium azide water and sodium carbonate, sodium bicarbonate, and sodium hydroxides. And it has the recycled materials, such as solvents, reagents, and catalysts, are confirmed source of nitrosamines. So these are all some of the root cause of nitrosamines in API manufacturing process. So moving on to the next section, small molecule nitrosamine risk factors in API. One of the risk factors for the formation of small molecule nitrosamine is the use of secondary amine or tertiary amine bases or tertiary amide solvents, and the nitrosating agents are deliberately added to the process, or if the nitrosating agent is present as a significant impurity in the other materials, especially if these precursors are used in the same or adjacent steps. The other risk factor for the formation of small molecule nitrosamines in API is presence of 1,1-disubstituted itrazines or their amines in the process. Because I said that 1,1-disubstituted itrazine can react with the molecular oxygen, and it will produce the nitrosamine impurity. How about the NDSRI risk factors in API? If the starting material, intermediate, related substances, or API have a secondary or tertiary amine functional group, and nitrosating agents are deliberately added to the process, are present as a significant impurity in other materials, especially if the precursors are in the same or adjacent steps. Suppose if the process used the fluid bed drying or jet milling process, and the API or counterion contains secondary amine, then there is a risk for NDSRI in API. If the activated carbon is used in the purification of the crude material, crude or final API, and the API contains, API or counterion contains secondary amine, then there is a risk of NDSRI in API. Recommendations to API manufacturers. Look at the API synthetic route holistically. Don't look at piece by piece when you do the risk assessment of the nitrosamines. Audit the starting materials, intermediate vendors. Understand the risk coming from the purchase materials. And if a risk of nitrosamine is identified, confirmatory test of batches should be conducted using sensitive and appropriately validated methods. If nitrosamines are detected, then perform a root cause analysis and implement changes to manufacturing process to prevent or reduce nitrosamines, add control wherever it is necessary, and report the implemented changes to FDA. Approaches for determining acceptable intake limit of NDSRI. To determine the acceptable intake limit, first you check the AI limit appears on the table 1 or table 2. You can find the the link for the I provided the link for table 1 and table 2. You can check that table and then first check those tables to see whether that particular NDSRI is listed in the table 1 or table 2. If AI limit cannot be found in the above tables, determine using CPCA to determine the acceptable intake limit using CPCA recommended in the rail guidance. If they are unable to meet their CPCA-based acceptable intake limit, they can use read across analysis from a surrogate to determine the acceptable intake limit, or they can submit the compound-specific toxicology data to determine the acceptable intake limit. Certain nitroso compounds, for example, N-nitrosoamide, N-nitrosoguanidine, and N-nitrosourea, the CPCA, these nitroso compounds are out of scope of CPCA. For these type of N-nitroso compound, alternate approaches such as read across analysis from a surrogate candidate or compound-specific toxicology data may be used to determine the acceptable intake limit. Nitrosamine control strategies. The ICH M7 provides 4 options to control the mutagenic impurities. The API manufacturers can use that 4 options as a control strategies, but it has to be the control strategies should be supported by the appropriate data and justification. Suppose if multiple nitrosamines are controlled in the specification and the total level of nitrosamines exceeds the acceptable intake limit of the most potent nitrosamine based on the MDT, the manufacturer should contact the agency. I provided the contact information in the final slide. You can contact the agency in case if the the total level of the nitrosamine exceeds the level of most potent nitrosamines based on MDT. Challenge question 1. Fluid bed drying or jet milling in the API manufacturing process has potential to form NDSRI when API contains primary amine, secondary amine, tertiary amine, or amide. The answer is secondary amine. Challenge question 2. Which of the following nitroso compound is out of scope of CPCA? N-nitrosamines, N-nitrosoamides, N-nitrosoguanidines, N-nitrosourea. all the above except A. The answer is all the above except A. In summary, nitrosamine formation may be a risk in API. Process understanding is essential to mitigate the risk. Build quality into the process to minimize or eliminate the nitrosamine formation. Always take a holistic approach when assessing the nitrosamine risk. If you have any questions related to nitrosamines in API, use cedar-opq-inquiries@fda.hhs.gov. We will get back to you with the answer. And with that, I thank everyone. If you have any questions, I'm happy to answer in the question and answer session. So I would like to invite the next speaker, David, to the podium. Thank you.
Hello, good morning, everyone. I'm David Awachiotu, and I'm SPQA with the Office of Pharmaceutical Quality, mainly involved in post-approval changes of supplements. And pharmaceutical quality, we all have this natural inclination towards quality products for use in our everyday lives, such as computers, cars, cell phones. And drugs are no different. Patients expect very safe and effective drugs from us. So pharmaceutical quality basically is to assure that every dose that gets to the patient is safe and effective, it's free of contamination and defects. It is what gives them that confidence in their next dose of medicine. So today I'll be talking about post-approval changes in complex generic drug products from the CMC and then product quality perspectives. Per the GDUFA 3 commitment letter, complex drug products are defined as any product with a complex active ingredient or complex route of delivery. Any product with a complex formulation such as liposomes, colloids, complex dosage forms, or even complex drug device combination products. For the purposes of today's presentation, I'll be focusing on prefilled synthetic peptides, on metered dose inhalers, and then transdermal systems, as well as dry powder inhalation products. Per the Code of Federal Regulation, it requires that FDA be notified of any changes made to conditions established. And I would very much like us to focus on the phrase condition established, as we'll come to that later. It is applicants' duty to notify the FDA about any change in each condition that is established in an approved application beyond the variations that are already provided for in the application. And the notice is required in the form of a supplement for change. It's required that the applicant clearly list all proposed changes in the cover letter to that supplement. We also require that the holder of the application to assess the effects of these changes before distributing a product made with a manufacturing change, which means that supporting data must be provided for those proposed changes. The Code of Federal Regulation also provides us with filing categories for these based on the risk. There's a reporting category based on whether the proposed change has a substantial, moderate, or minimal potential to have an adverse effect on product quality. And when we talk about product quality, it talks about the identity, strength, quality, purity, and potency of the drug product as it relates to the safety and effectiveness of the product. Based on these reporting categories, we have major changes, which means that these are changes that have the potential. The key word here is the potential to have adverse effect on drug product quality after effecting those changes. we usually call it the tail, and then we wait for FDA to assess what has been submitted to support those changes. Once approval is given, then the firm can go ahead with effecting those changes. And it's in the, it's called a prior approval supplement. We also have moderate changes. And with the moderate changes, we have the two. You said tell, wait, and do, which I call the CBE 30, which means that applicant notifies or tells the FDA that this is the change we want to make within 30 days. FDA is supposed to actually grant that change, the CBE 30, or if there are any issues, applicants will be notified that no, the change you want to, you're proposing, or the change you're proposing must be in a higher filing category than you have proposed, in which case it's elevated to a PAS. Or you have the CBE 0, where the change is moderate. It's just tell, do and tell. You do it and then you tell. Same with the minor changes, which is the annual reportable changes, where a firm actually reports those changes in its annual report. I've listed here some current post-approval guidances as it relates to some common dosage forms, such as immediate release, modified release, semi-solids. And then there's the post-approval changes for alternate testing and laboratory testing sites. Changes to, which is the general, which is not dose specific, the changes to approved NDAs and ANDAs. There's also the annual reportable changes guidance, as well as for drug substances, and then the comparability protocol. For complex generic drugs, due to the complexity in nature of either the drug product or the drug substance itself, some of these guidances appear either outdated or not applicable, not specific to that particular dosage form. And that is where the challenges come, in that it results in filing categories for some products that are not clearly defined. And then for such products that are not clearly defined, it results in overly conservative approaches to post-approval changes. There's therefore the need for transparency and clarity regarding the life cycle management of these products. Some common regulatory barriers that such complex generic drug products face include, confusion as to which filing category would they fall into, as well as the data requirements for such changes. For this reason, and due to the timeline, confusion in timelines, ICH has come out with some guidelines from Q8 through Q14 that actually deals with early stage of the product life cycle, as well as drug product development. Of importance is the IC. All are very important, but the forward here is that we want quality to be built into the drug product, rather than for quality to be by testing of the product after manufacturing of the product. So we have that quality by design. We're going to focus more on ICH-Q12, which talks about, as I said earlier, established conditions, as well as the post-approval changes management protocol. Established conditions are legally binding or conditions which require submission of a supplement if these are changed. So they form the elements of control strategy in an application based on the product and process parameters or facility and equipment operating conditions or in-process controls, finished product specifications, and associated methods and frequency that are necessary to assure process performance and product quality of the drug. So if any established condition is changed, it requires a submission of a supplement. Supporting information does not require regulatory submission if changed. And the extent of ECs vary based on the understanding of the drug product, as well as the process, the manufacturing process itself, characterization of the product, and then the firm's own developmental approach, and then the potential risk to product quality. Product and process understanding come from the development studies, platform knowledge that is built, or commercial experience. After established conditions are identified, the applicant may follow existing guidelines or regulations or as far as the reporting categories for any changes that are proposed, or they can propose an alternate reporting category, but based on thorough risk assessment as justification. The reporting category is always dependent on the potential risk to product quality, as I stated earlier. So the risk assessment should follow approaches described in the risk management protocol in ICH Q9. Consider overall control strategy and then possible concurrent changes. What this does is it increases transparency between industry and regulatory authorities, in that there's better design of the product as quality is built into the product right from the onset, with fewer problems in manufacturing, since decisions are made based on process understanding and risk mitigation. And for agency, there's improved quality information and required regulatory submissions, as well as the quality of the review. Post-approval changes management protocol. It's referred to in the US by the FDA as comparability protocol, and it provides predictability and transparency regarding the information that's required to support a CMC change and the submission required for that change. It may be submitted with the original application or as a standalone submission. Could be a prior approval supplement, and can be submitted to address one or more changes for a single product, or it may address one or more changes to be applied to multiple products. For example, if a company has, let's say, injectables and then wants to qualify, let's say, a stopper for the products, it can submit a comparability protocol in a higher filing category. And then once that protocol is approved, it can be applied to all the other products. It submits, as long as the FDA approves those proposed tests as well as conditions that are submitted, then a lower filing category can be submitted for the other products to use those. Should note that CMC change that will require supportive efficacy safety, that's clinical or non-clinical or human pharmacokinetic or pharmacodynamic data, is not suitable for inclusion in a PA CMP. The first step involves the submission of a written protocol with the proposed tests to be carried out for the proposed change. Usually that's the strategy, is to submit a PAS with the listed number of tests and conditions to be met. Once the FDA reviews these tests or strategy and approves it, the firm goes to the execution stage, where it generates the necessary data for the exhibit batch, as well as validation and verification reports and stability. And then step 2, once it's approved, is for the firm to submit these data in a lower filing category. So I have provided here a timeline where, for, let's say, the submission of a PAS, assuming it takes about 4 months for those comparability protocols to be reviewed and approved, the firm goes through the execution stage and then generates the necessary data, submits an exhibit batch with all the supporting information. It's submitted as at a lower filing category. So in that case, a CBE 30 submission, that is to tell, wait, and do. So within 30 days, the firm can implement the change as long as all the conditions or the results that are generated meet the acceptance criteria and conditions in protocol. They are met. Applicant submits the information to FDA. If the acceptance criteria and other conditions stated in the protocol are not met, then the change cannot be implemented using this approach. The advantages with this is there's increased clarity and standardization. There's increased manufacturing efficiency, reduced regulatory burden, reduced drug shortages, reduced time, as well as costs. Post-approval changes to drug device combination. I'm gonna have to run through this. The current guidance for industry changes to approved NDA, ANDA, only gives 2 examples for container closure or for devices. Which talks about the manufacturing site, as well as for the container closure change itself to be submitted as a PAS for these devices. Design control for MDIs, that's for metered dose inhaler as well as for dry powder inhalers, require a combination of product understanding, process understanding, as well as device understanding to gain a thorough understanding of these. Usually, drug device combination products is a split review between CDRH and then CDA or CBA. I have provided a correlation between dry powder inhaler or MDI components, mainly dry powder inhaler, of some of the critical quality attributes for the device constituents as well as the drug formulation constituents that go together to affect the final product quality of these devices. For the drug substance, assay particle size, as well as the morphic form of the drug substance, excipient CQAs, device CQAs, all go in together to affect the final drug product quality that is provided. And knowledge of these very helpful in effecting changes post-approval. The FDA came out with a guidance for the ICH implementation strategies for, and part of it includes the identification of established conditions and then the reporting categories for the device constituents part of a drug device combination product. So the 3 questions that he asked is whether the device constituent part is very essential for safe use based on risk management, or whether it's essential to achieve the delivery of the labeled dose. And does it impact drug product critical quality attributes? If the answer is yes, then the device constituent is actually very essential or performs a primary characteristic to the drug product. Based on the level of potential risk, either PAS or CB30 or CB0 can be submitted. If the answer is no to all, then it's not reported. Its annual report is submitted in the firm's annual report. Here's a challenge question. So if an applicant submits a CB30 to propose the extension of micronization pressure parameter range during the micronization of the drug substance that was used or that's used in the manufacture of a dry powder inhalation product. Applicant cites section 7C1B of the post-approval changes guidance. That's a change in drug substance manufacture or parameters to support the proposed change. Should this supplement be granted CBE30 based on the submission, or should it be elevated to PAS based on section 7B1 of the drug post-approval changes guidance? Because the changes in the micronization pressure parameters involve elevating the upper limit of those parameters for the drug substance. And the answer is B. It should be elevated to PAS. And the reason is that the revision to the drug substance micronization process parameters may affect the particle size distribution for the drug product. We all know that one critical quality attribute is the aerodynamic particle size distribution for a dry powder inhaler. So it will be elevated to PAS based on that, the criticality of that change. Post-approval changes to transdermal delivery systems. We know that transdermal delivery systems usually consist of the drug and adhesive mixture, the release liner, the backend, based on whether it's a matrix type transdermal or it's a reservoir transdermal or topical delivery. The questions remain as to what should be submitted. How far should the firm go into the characterization of any change? Is there a BE study that should be performed based on that change, or in vitro skin permeation test, or in vitro release will suffice it? That is where the confusion is. I've provided some supplements where the changes have been submitted as a PES or a CBE 30 for examples of these. The challenge question is, an applicant submits a CBE 30 to propose an increase in batch size less than 10 times, changes to components of the adhesive and source of foil within the pouch material. The data submitted to support the proposed change indicate that the changes do not adversely affect draft product quality. Should a supplement be downgraded to a CBE 0 based on the data provided? Or should it be granted as a CBE 30 as submitted, or elevated to PAS due to the potential high-risk change to the adhesive component? And the answer is that this is a multiple related change, so it's elevated to a PAS. We know that the adhesive or adhesive component represents a change in the component of a formulation, and it has a substantial potential to have an adverse effect on product quality. The adhesive of a transdermal delivery system is critical to the safety, efficacy, and quality of the drug product, since it is in intimate contact with the drug substance and or other excipients that may alter either the adhesive properties and or influence the release of the drug. So the key word here is the potential of that change to have an adverse effect on product quality, and not how good the data looks. So synthetic drug products, the FDA defines peptides as any alpha amino acid polymer composed of 40 or fewer amino acids. FDA released the guidance for submission for NDAs that refer to RLDs, or recombinant DNA origin. Due to the current state of technology for peptide synthesis and characterization, applicants can submit ANDAs under the 505J pathway of the Food, Drug, and Cosmetic Act, for which synthetic drug products that reference previously approved peptide drug products of recombinant DNA origin. So generic peptide drugs must demonstrate the sameness to RLD, in that the API must be characterized to show that peptide-related impurities are same or lower than those of the RLD, and any new specified peptide-related impurity that is found must be less than 0.5, and the appropriate justification provided on why or how its presence would not affect the safety and effectiveness of the generic product compared to the RLD. So in the absence of a specific post-approval changes guidance for generic synthetic products, post-approval changes have been based on a combination of risk assessment and knowledge gained from the original submission. And especially, I'm going to hit especially on for approved drug substance. Any change in the drug substance manufacturing process require the applicant to demonstrate that the new peptide is thoroughly characterized to show all peptide-related impurities are the same or lower than the approved drug substance, as well as the RLD. And to characterize any new specified peptide-related impurities and justify why the presence of such new impurity would not be expected to affect the safety and effectiveness of the drug product. So characterization of the generic drug substance, provide adequate information on the primary sequence, secondary structure if there is, oligo or aggregation studies, as well as the biological activities. These are examples of some PAS and CBE30s based on the type of changes that are proposed by the applicants. So in conclusion, it's applicants' responsibility to improve the quality of submission by using science-based and risk-based approach to assess the impact of proposed changes on drug product quality. To also demonstrate good product and process understanding of your supplement. That's, you should adopt modern quality techniques with focus on sound science for assessing and mitigating risk of poor product quality and process quality. So quality by design, CQA, CPPs, CMAs, and control strategy needed. So at the end of the day, we'll be able to stand by our products. Thank you.
Thanks, David, and thanks to SBIA organizers for this opportunity. My name is Asif Rashid. I'm a senior review chemist in the Office of Pharmaceutical Quality Assessment 2 within OPQ. And today I'm going to talk about this guidance for industry, rather the draft guidance for industry, which is titled Quality Considerations for Topical Ophthalmic Drug Products. Oops. And for those of you who are attending this event for CME credits, the learning objective for my presentation is to provide you with an overview of the draft guidance, and explain current practices on assessment of key topics discussed in the guidance. This guidance covers ophthalmic drug products which are intended to topical delivery only. And that may include gels, ointments, creams, liquid formulations such as solutions, suspensions, and emulsions, and the applications under which these dosage forms are submitted. And these are NDAs, ANDAs, BLAs, and OTCs. The guidance was initially published in October of 2023 and subsequently revised in December of 2023 to add one more technical section. Currently, as of now, the guidance has 7 substantive technical sections, and these are listed here. These include microbiological considerations, visible particulate matter, extractables and leachables, impurities and degradation products, in vitro drug release and dissolution testing for quality, container closure systems, design and delivery and dispensing characteristics, and finally, stability. So I'm going to briefly discuss each of these sections and just highlight the salient features and expectation for these sections. So the guidance provides that for microbiological considerations related to product sterility for all ophthalmic drug products. And emphasis is placed on complying with current good manufacturing requirements to ensure product quality. And in that regard, the guidance refers to 2 documents. The first is another guidance for industry, and that is sterile drug product produced by aseptic processing, current good manufacturing practices. And the second document is the submission documentation for sterilization process validation in applications for human and veterinary drug products. The guidance puts specific consideration for multi-dose drug products, as you can imagine. For multi-dose drug products, because of multiple use, the susceptibility for microbial ingress is significantly high. Yet, these products should remain free from any harmful contamination if such a potential microbial ingress happens. So due to this concern, the guidance recommends use of preservative if the drug product itself does not have inherent antimicrobial activity. And speaking of preservative, lately some applicants have approached the agency with proposal to use silver sulfate or other silver-containing compounds as preservative, and agency has not recommended that because of safety concern. Some applicants have approached agency with multi-dose preservative-free formulations, and for those type of formulations, the guidance recommends applicants that they must demonstrate protection, robust protection for each unit from incidental contamination during multiple use, as well as provide validation of all aseptic and sterilization processes. The next substantive section is visible particulate matter. And in general, testing for visible particulate matter is accomplished by visual inspection, and this is to ensure that the products are not adulterated. However, we know that most ophthalmic dosage forms, the liquid, they are packaged in opaque containers, and for that, applicants can use either a destructive or a non-destructive testing. And I should point out that the guidance is silent on what type of destructive or non-destructive technique could be used. So that is up to the applicants. Applicant can choose a technique of their choice and provide adequate information to demonstrate, to justify the suitability of that technique for the intended use. For dosage forms, where the particulate matter are by design, for instance, suspensions and emulsions, stability testing could be or should be used to evaluate changes in particle size over the shelf life. And this is done to accomplish, to ensure that any intrinsic particulate matter should not be there due to product instability. For extractable and leachables recommendation, the guidance expect that the assessment for extractables and leachables for primary, secondary, as well as tertiary packaging components, including labeling of the container closure system. And the studies for extractables and leachables should be performed within the framework of the USP General Chapter 1663, which is the assessment of extractables associated with pharmaceutical packaging and delivery systems, as well as USP 1664, which is the assessment of drug product leachables associated with pharmaceutical packaging and delivery systems, and this guidance for industry. Container closure systems for packaging human drugs and biologics, chemistry, manufacturing, and control documentation. So I'm not going to go into the details of the methodology and the requirements, because after lunch, there's a whole presentation on this topic alone. So you are going to get more insights from our colleagues on the topic of extractables and leachables. Specific for leachable study, the guidance provides the safety thresholds. And these are given as follows: 1 ppm for reporting threshold, 10 ppm for identification, and 20 ppm for qualification threshold. And here I would like to point out that these numbers should not come as very new to our colleagues in the industry. These numbers have been out in public domain for quite many years. It's just that now the guidance has formalized them. And we believe that there is merit for this approach. Providing these thresholds upfront takes some level of uncertainty out. So if you are a generic applicant, you are working on your leachable study and your stability study, you are monitoring the leachables, so you know when to take action for identification or qualification should a certain threshold is reached. And also, leachable testing should be performed by fully validated analytical methods. And just a point of caution that the limit of quantitation for the analytical methods should be less than or equal to the identification threshold. The next substantive section for the draft guidance is impurities and degradation product. So in general, NDA and ANDA drugs follow ICH Q3B. OTC monographs are generally covered under USP 1086. But for NDAs and ANDAs, the information on impurities are provided along the lines of these 4 bullets. So it's for each specified identified degradation product or impurity, each specified unidentified degradation product or impurity, any individual unspecified degradation product or impurity, and total degradation products or impurity. However, this section provides a footnote which states that acceptance criteria for specified degradation products in generic drug products should be established according to the guidance for industry and as impurities in drug products, which was published in November 2010. So that means if you are an applicant for a topical ophthalmic drug product, for a generic, rather, topical ophthalmic drug product, you should be following that guidance in order to establish a specification criteria for a specified degradation product. But this guidance provides specific recommendation for unspecified degradation products or impurities, and those thresholds are different than the corresponding thresholds in ICHQ-3B. And the guidance gives you the rationale. Why is it so? For one, that these impurities poses potential for high local concentration in the eye, and there's a lack of data on potential effects for these impurities. So because of that, the thresholds given are different from ICHQ-3B, and they are given in this table. So you may notice that the thresholds are two-tiered. For drug products with strengths greater than 0.1% to less than or equal to 1%, the recommended identification and qualification threshold is 0.1%. However, for drug product with lower strengths less than or equal to 0.1%, the recommended threshold is 1% or 1 ppm. That is 1% of API or 1 ppm of drug product. For drug product with strengths higher than 1%, the recommended identification and qualification thresholds would be considered on a case-by-case basis. And also please note that these thresholds are for both identification as well as for qualification. That means if you observe an impurity, an unspecified impurity above these thresholds, we expect the applicants to identify as well as qualify these impurities and provide the safety information. We should address both local ocular toxicity as well as general systemic toxicity, and those information will be assessed by relevant disciplines within the agency. The next section in the guidance is in vitro drug release / dissolution. The guidance recognizes that rate and extent of drug release is a quality criteria to ensure consistency for certain dosage forms, namely suspensions, emulsions, and semi-solids. And for that matter, this test could be used or should be used as part of quality control strategy. And the guidance provides an alternate also. So if an applicant believes that there are one or more critical quality attributes that are sensitive to the formulation and process variants, then those CQAs could be used in lieu of in vitro drug release or dissolution testing. With this, time for a challenge question. For leachable study in topical ophthalmic drug products, thresholds at or above which the observed leachable should be qualified for safety is, I'll give you a few seconds to think. And by show of hand, if you believe it's A, 1 ppm, or 10 ppm, or C, 20 ppm, or D, 0.1%. I think most of you agree it's 20 ppm, and you are absolutely correct. So moving on with the guidance, the next substantive section is the container closure design and delivery and dispensing characteristics. And this section has multiple subsections. The first subsection is tamper-evident packaging. And this is specifically for non-retaining tamper-evident ring. As depicted in this picture, that in the past, agency has received multiple complaints from consumers that the non-retaining tamper-evident, sometimes it is loose enough that it slips past the bottleneck. And if a patient is going to administer the drug, that loose ring can fall off on the eye and causes injury. And that's not acceptable. So the guidance recommends that applicants should employ some sort of positive retention mechanism to ensure that the ring stays in its place and does not fall off. The next subsection is for the container closure tip, and specifically the tip which are sealed, and they are sort of unsealed only at the time when the patient first time uses the container. And if that procedure involves multiple steps, then the agency discouraged this practice. So for instance, if the package insert instructions tells a patient to twist the cap clockwise, and that motion, because of a spike inside the cap, pokes a hole on the tip. And then the instruction says to the patient to twist the cap counterclockwise to open the container and then administer the drug. We find that this instruction could be complex to certain consumers and may lead to confusion. And we have received reports that some consumers or some patients were not able to appropriately puncture the hole on the tip, and then they resort to certain other techniques. They start using knives or scissors, and that's a huge risk for contamination. So that is discouraged. Guidance recommends that keep it simple. Simple instruction, use of single-step procedure with simple directions. The next subsection is for torque specification, or the twisting force that is required to open the container. And there the guidance recommends for the applicants to consider the patient population. The torque should be low enough for elderly patient population to open the caps without difficulty, and it should be high enough that it remains in place during manufacturing, storage, shipping, and handling. Regarding the color coding for caps, which is to characterize the therapeutic class, guidance recommend to use the uniform color coding system as described in the American Academy of Ophthalmology's color codes for topical ocular medications policy statement. For the delivery and dispensing characteristics, the guidance provides the fill sizes for unit dose containers. So for liquids, non-preserved liquids, solutions, suspensions, and emulsions, the fill volume should not be more than 0.5 ml. And for semi-solids, ointment, and gels, the fill size should not be more than 1 gram. And these unit dose containers should not be able to recap. So it's a one-time use. Any leftover drug should be discarded. For multi-dose containers, the guidance provides a recommendation for drop size, and it should be between 20 and 70 microliters. However, for generic drug products submitted under ANDAs, the drop size should also be within plus minus 10% of the drop size of the reference listed drug, and be within the drop size of 20 and 70 microliters. However, if there is deviation, then applicant can justify those deviations to demonstrate similar number of delivered doses. Then the next subsection deals with suspensions, specifically suspensions which are packaged in multi-dose containers. So a quality test for resuspendibility or redispersibility should be proposed in drug product specification, and this is a qualitative test, and it should be performed per labeling instruction to mimic actual patient use conditions. So for instance, if the labeling instruction says to shake the bottle before use, right? So you can anticipate what could be the average time a patient is going to shake the bottle, right? And based on that, you can propose some acceptance criteria. So for example, just an example, the acceptance criteria could look like that time for resuspension for drug product after storage could be between 15 and 30 seconds. Then also for suspensions packaged in multi-dose container, test for dose uniformity should be proposed, but that's only a one-time study. And that's a quantitative test. It is performed by withdrawing the drug from the top, middle, and bottom of the container after shaking per labeling instruction. And then the samples should be tested by assay, for assay using assay acceptance criteria. So this test ensures that throughout the container, all the doses are uniform. Switching to the last substantive section of the guidance, that's stability. And in general, stability studies are to evaluate the appropriate storage conditions and the expiration date. So for instance, the ophthalmic drug products packaged in semi-permeable containers, for example, low-density polyethylene, they should be kept at relatively low humidity conditions. And those low relative humidity conditions are provided in ICH Q1A. But Applicants have choice to use higher relative humidity. It's just that they can use the calculation provided in ICHQ-1A to derive water loss at the reference relative humidity. Then the section provides recommendation on orientation during storage. And for NDAs, preliminary development work can be done to evaluate the storage conditions in two different orientations, so upright and inverted or horizontal. And then the worst case orientation for batches should be used for batches representing commercial manufacturing processes. For ANDAs, however, two different orientations are tested for the primary stability batches. And the data from both orientations should be provided and analyzed. And the worst case orientation is used for routine stability testing. For BLAs, the primary stability studies are performed to determine the storage conditions under real-time conditions. Then the guidance provide recommendations on certain studies. So for instance, study for water loss should be conducted to assess moisture transmission properties of the CCS and protective properties of any secondary packaging. Freeze-thaw study should be done for emulsions and suspensions. It's a one-time study, and guidance provides condition that it should be 3 cycles from freezing all the way back to ambient condition for a cumulative minimum of 3 days. However, applicants can justify alternative conditions and durations with adequate justification. In-use stability is performed if the labeling claims has some indication that the storage condition may change after opening, such as change in temperature or change in light exposure. With this, time for a second challenge question. Which of the following studies is most suitable to mitigate the risk to drug product quality for ophthalmic suspensions and emulsions due to temperature variations during shipping and storage? Give you a moment to think, and again, by show of hands. Could it be in-use stability or photo stability, water loss study, or freeze-thaw? Yeah, if you are thinking it's freeze-thaw, you're absolutely correct. So that concludes my presentation, and I'm going to sort of, as a take-home, just point you to the last bullet, that adequate supporting data in ANDAs and application that ensure a smooth and timely assessment of applications and ultimate approval. And this guidance hope to accomplish the same. And with this, I am going to hand over to Brenda.
Thank you very much, Dr. Rashid. Don't go anywhere. We invite the other 2 speakers to come back up to the podium, please. Time for Q&As. Okay, everyone is familiar with the process. Do we have any in-house questions? Okay, do we have any online questions?
Sure. First question for Kumaran. Do you see the duplex sequencing as an acceptable method to determine the safety of NDSRI instead of TGR?
Could you please repeat the question, please?
Sure. Do you see the duplex sequencing as an acceptable method to determine the safety of NDSRI instead of TGR?
Okay, that's a good question, actually. So the NDSRIs, so if they don't use the CPCA-based acceptable intake limit, then the agency has proposed 2 alternate approaches. That is the surrogate analysis, the read-across analysis using surrogate candidate, and then the compound-specific toxicology data. They can submit the compound-specific toxicology data to determine the acceptable intake limit. But the rail guidance, which is published in August 2023, they provided some information about what to submit in the compound-specific toxicology data. So I think the question is that they should refer that the rail guidance for NDSRI. So they provided some information about the the compound-specific data regarding that particular toxicology studies, what they are saying. Actually, I am a CMC reviewer. This is a farm tox question, so it's out of scope of the CMC, out of scope of my knowledge. So I think at least to get some preliminary information, they should refer that rail guidance. It has some information about the compound-specific toxicology data, what to submit, what not to submit.
Thank you. First question in-house.
This is for Asif Rasid. For the acceptable leachable of the ophthalmic drug product, it is recommended to have a labeling also included in the acceptable leachable study. So if a product is using package insert, which is not directly like it is on the cap of the bottle, do we need to include that in the study too?
So in ideality, you try to mimic as close to the commercial packaging as possible, right? I understand that maybe at the time of development, you may not have developed the entire tertiary packaging system, but there are ways to circumvent around. And the package insert, it's part of the label, right? So I would say that yes, it should be part of the tertiary packaging system, which should be put in place for leachable study.
Okay, thank you.
Thank you. And our second in-house question, please.
Good day. Thank you again for all your presentations. So can the agency comment on how successful the comparability protocol has been? For example, how many have been approved versus submitted? And have you seen success in reducing regulatory burden?
You can turn on the mic, please. Yeah.
Yeah, it's been very successful. It cuts the timeline for submission. As to the numbers that have been approved, there is, I would refer that to, there's a comparability protocol team. I don't have the numbers in front of me, so I can't tell. But as far as I can tell, it's been very good. And it actually gives more information to the FDA when it comes to the approval process for post-approval changes.
Thank you.
Question, but I'll wait.
Okay, you stay right there then.
Yeah.
Question from online, please.
Another question for David. Will the FDA publish a guidance for industry for post-approval changes for complex generics in the near future, as the current guidances may not be applicable to these specific complex drug products?
For questions about guidance, I would refer you to the, there's a website on the FDA, that talks about guidances that are in line to be released. So I wouldn't be able to give you a specific answer as to, yeah. But I would also use this opportunity to say that if you have any questions or if you have any doubts with the submission for these complex generic products, controlled correspondences are your friends. You can submit controlled correspondences and then you receive confirmation from the FDA on what to submit or the filing category that is needed. Yeah.
Thank you. And just for informational purposes, every year for the past 4 years or so, we've been holding a complex generics 2-day conference in September. And we will be working on doing one this year as well. If you go to SBIA website, business cards over there, you will see all the webcasts from the past few years of complex generic drugs. It's called Advancing Generic Drug Development in the past few years. So please take advantage of that wealth of resources that we do have already available. Thank you. Question in-house, please.
Hello again. This is kind of related to the last question, but a little more specific. Does FDA intend to update the changes guidance or any of them to address, for example, the question that you posed several times of number of batches and things such as that, which are somewhat addressed in SUPAC, but obviously that's a very old guidance.
As I already said, guidances has to do with, I think, policy, and then OPPQ, I think. So I wouldn't, but what I would say as a CMC reviewer and for post-approval changes is that control correspondences with such questions are the best way to go. And then based on FDA's current thinking, we respond to those. It actually even helps to gather more information if there is any update as to what to put in, and then the way forward. So again, I wouldn't be able to give you a specific answer, but control correspondence are the way to go. Such question.
Thank you. Do we have a second question in-house?
So this is a in general question. I think it's a good panel for impurity questions. So I notice a lot of times you'll see ICH guideline when you calculate the limit is one thing and USP is another. And then you go to the FDA's guidance for lack of proper justification for impurities, and one of the footnotes in center, you have to use the worst case, right? If it's ICH or USP, right? So I'm just wondering, is the FDA planning on having some sort of meetings or correlation with the USP to try to align the specifications with the compendia? So there's not inconsistencies, because I can imagine that it would reduce the number of post-approval supplements. Just a thought.
I can start and then my colleagues can chime in. This is a great question, and unfortunately, I'm not able to answer it right now. So I'll just leave it at that.
No more comments?
So the USP as is a gold standard for all these limits for all these impurities. So of course that is the minimum requirement when you want to control some impurities. But also if there is some specific issues are there on the on the impurities, then we would follow the ICH guidelines. For example, any unspecified impurities, the ICH guideline says that it is 0.10%. So we just follow that limit as a standard impurity control for the any unspecified impurities. So of course the USP also we take into consideration, but when it comes to controlling certain specific impurities, we also take into consideration of ICH guidance too.
Thank you. Let's take a question online, please.
Question for Asif. For particulate matter for suspensions, you mentioned to check for particle size distribution changes over stability. Since the product inherently contains particles, are we exempt from testing for visible particulates in the drug product as a release testing?
So I would not say that applicants are exempt. USP Chapter 790 and then the Journal Chapter 1790 provides certain strategies for those type of dosage forms to test for particulate matter. The example that I provided in my presentation is for intrinsic particulate matter. That means how the interaction of the particles, the EPI interacting with the environment, the surrounding, and the container closure system, and if that indicates any product instability. For extrinsic particulate matter, I think USP 1790 has certain recommendations. And if there are compelling challenges to fulfill particulate matter requirement, and so that could be a good question for a control correspondence for us to see beforehand. Thank you.
In-house question, please.
This question is for David. When it comes to like post-approval change management control protocols, especially for multiple products, which are using like similar container closures, like we're using the same stopper, can there be like a tool where we can use like a group submission instead of making like separate submissions for each of the products?
Yes, group submissions are allowed in, and it has to be stated as long as your comparability protocol is approved. You can state as part of those the submission that if this is approved, maybe it's going to be a lower filing category for some products. As long as your test and every conditions that you specify in the comparability protocols are reviewed and approved, then a group submission can be done. There are instances where group submissions have been done for a lot of, let's say for like stoppers or the same container closure for the other products.
Okay, but that has to be submitted through like a normal ECDD, not like to seed or next gen or something.
Can you repeat what you said?
That can only be submitted through like normal ESG gateway, not through like a seed or next gen portal, right?
Yes. Yeah, group submissions can be made through the ECTD.
Okay, thank you.
All righty, thank you. Online question, please.
Question for Kumaran. Does the API manufacturer need to submit risk assessment documents to the agency?
Actually, they don't have to submit the risk assessment documents to the, in the DMF submission, but it should be available whenever it is needed. It should be available at the site for the evaluation.
Next online question, please. Thank you.
Another question for Asif. For a freeze-thaw study, if the RLD label doesn't mention do not freeze, but the generic product fails any CQA on a 3 freeze-thaw cycle, but okay for 1 or 2 cycle, does the generic product label need any special note? Is that acceptable, or do we need to test the RLD and show that the RLD has the same issues?
I think that's a little bit of a specific question. Without knowing the details and the data for that particular product, it would be a little difficult to answer. So my recommendation again, if the questioner has a specific case in mind, so that could be submitted via controlled correspondence, because that needs to be looked at what's going on in terms of product quality, and if the change, if the labeling has to mention certain instruction in terms of do not freeze, for example. So that would be a deviation from the RLD labeling. So that's a multi-pronged question, and I think it is best to be addressed via control.
Thank you.
Can I also address the last question about the group submission for comparability protocol? I would also like to state that, yes, group submissions can be, it can be submitted to the FDA, but it has to be approved as a group submission before it can continue. And as usual, one of the conditions for group submissions to be accepted or approved is that the same piece of information should be submitted for all the groups involved. So once that is met, yes, you can propose a group submission, and then once it's approved by the FDA, it can be reviewed as such. Thank you.
Thank you for that answer. I think that particular inquirer is out of the room, but thank you for that answer. It will be on record. Thank you. Online question, please.
Okay, another question for David. Can an applicant submit a controlled correspondence to seek clarity on post-approval change data requirements while the original ANDA is still under review?
Can you repeat the question again?
Yes. All right.
Let's wait 1 second while the drilling stops. Not exactly the music we're looking for. Okay, let's try again, please.
Okay. Can an applicant submit a controlled correspondence to seek clarity on post-approval change data while requirements, or change data while the original ANDA is still under review?
Did you hear the entire?
Yes, I did. And the answer is yes. But as part of the submission for the controlled correspondence, the applicant has to state that the submission is currently under review. And then you state the changes that you, or the clarification that you are seeking.
Thank you. Okay, next question,
One
please.
more online question. Okay, for Asif, does your presentation contents apply to ODIC preparation also?
Could you please repeat?
Does your presentation contents apply to ODIC preparation also?
OTIC, yes.
So yeah, in general, there are similarities between OTIC and ophthalmic. This particular guidance and my presentation is specifically for topical ophthalmics. So it should be taken as only for topical ophthalmics.
Thank you. Any more online?
Yeah, one more question for David.
If you could try to
Sure.
speak a little louder than the drill, we'll be okay.
When multiple related changes have different recommended reporting categories, should the submission adhere to the most restrictive category as suggested by CEDAR?
The answer is yes. It's the most restrictive filing category that carries the day at the end of the day for multiple related changes.
Thank you. How about you? Any more online questions? No? Any in-house questions? Going once, twice. Okay, let's thank our panelists and our speakers.